All right, guys, so we have now talked about two possibly electrophoresis lab experiments. One was on a color blob that was just going to take you through the motions of comparing the lanes in electrophoresis and trying to figure out what is present, what's not, and this concept of parents. So half the mother, half the father goes to the children, and we can identify the child's parents based on that. The next one that that we've already talked about probably is the crime scene evaluation. And that's what you're seeing there on the screen. It was this idea that we had a couple of different lanes, one DNA sample from a crime scene, and three suspect DNAs. And we were after a perfect match in that scenario because we're trying to determine if that person was present at the scene of the crime. It's a yes or no, so therefore their DNA should be present. And it should be a perfect match if it is. This one we're going to talk about again in terms of mother and father. And the reason I do this a second time is because this one does not use color blobs. It uses the traditional gels and the banding that you would see in gels. So it's a little bit more true to real life, I guess you could say that. And we're going to go through the motions just like we did with the other particular gels that we've ran and that we've talked about so far. So here's the overall view of the banding that's happened in the DNA paternity test lab experiment that we have done together. And up at the very top, you're going to see in the header, it says, whoa, wait a minute, there's missing bands. So there are missing bands in the gel, missing as in they're not showing up very well in the pictures. So I'm going to have to somewhat draw them on there for you so that way you will recognize that they are present. I think this picture, if you look at the DNA ladder, you can see 1, 2, 3, 4, 5, and maybe 6 bands down at the bottom, but there's actually 7. And then as we go through each individual lane, you're going to see that as well. So I'm going to talk about these missing bands and I'm going to draw them if you can't necessarily see them. So, in the very first one that we are going to talk about, this is lane number 1, and this was the ladder that we injected into that well. So the ladder is going to have a different assortment of base pairs. Those base pairs are going to separate. Those base pairs are going to travel through the gel, and we can measure the distance of those base pair bands. And that's why I'm holding up a ruler here, of course. So most of the time when I do the distance, I am actually looking at the top part of the gel and I am tracking how far down that band has traveled. So I'm going to let you go through the motions, maybe pause the video and take a look at the ruler here and figure out, okay, what bands have traveled where, and record those distances in terms of centimeters. There are a total of 7. So 1, 2, 3. This is number 4 that you're seeing there. I think that shows up pretty well. And then down here, these were very faint, or it almost looked like in some of the pictures that I was taking, it almost looked like it was just a big smudge. But there are 3 separate bands that's happening down here at the very bottom. So there are a total of 7 base pair fragments that you need for lane number 1. So, as before, go through, write down the centimeters, because you know as good as well as I do, there's going to be a graph, and distance is going to be part of that graphing feature. So, centimeters, perfectly okay. I also want you to keep in mind that based on the wells, where this ruler stops, I mean, nothing should go below 5 centimeters, right? Nothing will go below 5 centimeters. And the reason I'm mentioning that now is because in lanes upcoming, this ruler will go out of focus. I didn't do a very good job taking pictures with this one, and you're going to see it's going to be a little bit harder to pinpoint distances here because of that. All right. Next up is going to be lane number 2, and lane number 2 was a mother lane. So this was the mother that was injected, and you can see there are 2 bands that show up in the mother lane. The one at the very bottom I drew, but it was present. Again, it just doesn't show up very well on the picture that I submitted within PowerPoint or maybe on your computer monitor. So, again, record those distances for each band because each one of those are going to be important. Next up is the child band. So here is a child band or a child lane. There are a total of multiple bands here. And if you take a look, it looks like a total of 4. So hint, hint, there's a total of 4 there. I think 2 of them you can see pretty well, and the other 2 that are down below, I drew on there for you, so that way you can see those, and those are a little bit more evident. I believe that probably what's throwing you off, depending on how bright your monitor is a band that is right there. So there is a band that shows up. It shows up in the picture. It's present, I promise. But depending on your monitor and your adjustments, that might be a little harder to see. So a total of 4 bands for that particular child, right? Next up is going to be father number 1, and father number 1, a possible father, is going to show up in this lane, and this is where my pictures get a little fuzzy. So keep in mind, the gel is not longer than 5 centimeters, all right? So we're going to use that as a judgment call to go by. And then this particular band, it looks like there are 3 bands. So in father 1 lane, we see a band, of course, that's there. This on the, let me change the color of my marker maybe. So that one might be a little out of focus, but that is our 3 centimeter line, and that's 3.1, 3.2, 3.3, 3.4, 3.5 is right here. Then 3.6, 3.7, 3.8, 3.9, and then a 4 is right there. And then I think that you can somewhat see the tick marks after that point. So 4.1, and then 4.2, then 4.3, then 4.4, 4.5, 4.6, 4.7, 4.8, 4.9, and then finally a 5. So that is the ruler scale, and that maybe will help you determine what length of the bands that these have migrated to. So there are 3 bands in total. 1, 2, 3. 1 I've had to draw on the gel because, again, it was very faint. And the other ones, maybe not so much. They look pretty good, at least from my computer screen, and hopefully yours too. Next up is father number 2 lane, and in father number 2 lane, there was just really 1 band here, nothing more. That band does show up, but God almighty, this is like way out of focus, so there's the band. And I got to go through the motions of kind of telling you where these numbers are going to be. So right down here, this is going to be our 5 mark. And the 5 mark, of course, is off of the gel, so we know that anything that is 5 or greater is not a valid number, because the gel stops at like 4 point something. It doesn't even go to 5 centimeters. And then if I backtrack here, well, this looks like that's a possible of a 4, and then this looks like a possible of a 3, and there's our 1, or our 2, sorry, and then our 1 is up there. So kind of use that as a judgment call here. This is going to be a 3, and there's a 3.1 and 3.2 and 3.3 and 3.4 and 3.5, and then 3.6, 3.7, 3.8, 3.9, and then a total of 4. So there's kind of the out of focus description of what's going on, and hopefully you can use that very brief discussion to figure out what the proper distance is for that particular band. So overall, again, just a backward view of the unmarked gel. This is kind of what's showing up here. But keep in mind, there are missing bands here, and those missing bands are going to be important, so you'll need to use those as a judgment call to figure out what's going on. So as with the color blobs, your goal here is to say, all right, well, we have a child, and we only have 1 child that we ran in this particular lab experiment. And that 1 child, we have to determine who is the mother and who is the father. So we've got 2 different mother choices, mother 1 and mother 2. And the question here is which one shares half of those bands. So based on the child's banding pattern, half of those will come from the mother. So which one of these mothers, if either one of them, which one of these mothers would be the best fit choice? And then finally, we're looking at two different fathers, father 1 and father 2. And the same thing's going to happen here as well. You need to take a look at the two different banding patterns of the fathers and figure out which one of those is matching half to the child. So maybe there's one, maybe there's none. So who knows? That's just the thing. So there's kind of the rundown of the DNA paternity gel that we analyzed and that we ran. So again, once more, it is half and half. So there's just another picture of the overall view of the gel with unmarked bands that are not showing up very well on your side. And then here, yet again, is the rundown of the different types of samples that we analyze. So lane number 1 is the DNA ladder. We've got a total of 7. And then lane number 2 is the mother. Then lane number 3 is the child. Lane number 4 is father 1, and lane number 5 is father 2. So I guess I misspoke. I was thinking about another lab here. So with this particular band, there's the ladder, then the mother, then the child, then father 1 and father 2. So the child doesn't really have two mothers that we ran, not with this one at least. In another packet that we have, it does. But this one, we just have one mother, and that is it. So we identify the bands that that mother is bringing to the child, and then the other bands that the child has, where are those coming from? Is it coming from father 1? Is it coming from father 2? Or maybe neither one of them. Maybe we haven't picked the right potential father, and maybe we need to look at a different type of suspect in order to run those particular samples of DNA with. All right, so with that said, that's really all that I need to talk about the DNA paternity test. Once more, go through the pre-lab video, figure out what this is all about. I give you some description. Just as before, what you're going to find out is that the data sheet is going to make you plot a curve, and plotting a curve means that you're going to have the number of base pairs in some form or fashion, as well as the distance, typically reported in centimeter, on the Excel graph that you're generating. As long as you plot them the proper way, keep in mind, how do we plot them? What goes on the X? What goes on the Y? Is it straight up centimeter? Is it straight up number of base pairs? That's something that you can find out in our lecture videos. It's also something that you can find out maybe in our pre-lab videos. But either way, you're going to get a regression line. A straight line is going to come from this. And that straight line is going to be Y equals MX plus B. So, based on the distance that these bands have traveled, based on the distance that the bands have traveled, you are going to plug that in appropriately. And when you do that, you can solve for the number of base pairs with each one of those fragments from each one of those lanes. And that's what this lab data sheet is going to be all about. So with that said, good luck, folks. Again, review the graphing, review how we solve for the number of base pairs algebraically, not just estimation wise, and good luck. So if you need some help on some of the electrophoresis stuff, feel free to let me know. That's why I'm here for you. So send me an email, send me in class, come to the office. I'll be glad to kind of sit you down and talk about some of the problems that you're having. So hopefully you've learned something with the electrophoresis labs, and there could be more. So just stay tuned.